Hello everyone, this is Valentin Couvreur from the University of Louvain in Belgium. I'm excited to be here with you today to tell you about the work of my colleagues and I on water flow across roots. In this presentation, we address the long-standing enigma of water uptake uphill of water potential gradients observed experimentally and propose another theoretical solution to it. As a starter, I would like to give you an overview of what drives radial water flow across root tissues. And first, you need to know that passive water flow always occurs from high to low water potential. Across non-selective paths, such as cell walls, here represented in gray, but also plasmodesmata, water flows from high to low pressure potentials in blue. However, when you have a membrane, like here, the affinity of water for solutes comes into play under the form of the osmotic potential, which is negative and becomes more negative with larger solute concentrations. This osmotic potential is summed up to the pressure potential to yield the total potential, and water flows downhill of total potentials across the membrane. Then how do plants generate positive pressures in xylem vessels which do not have any membrane? For that, they created a very smart system based on a scaffold that you can see here in red, the apoplastic barrier, which limits the flow of water and solutes across the endodermis layer. Solutes may now accumulate in the xylem, where the osmotic potential becomes very negative, and so does the total water potential. This facilitates the uptake of water through the transmembrane pathway, but also the buildup of water pressure to positive or less negative values. In case there is a breach in the apoplastic barrier, then there is an extra pathway for water through the cell walls downhill of pressure potentials. This configuration is very similar to the one that you can see here on the left for the simple membrane, and that is why root radial water flow is often considered as analog to water flow across a simple big membrane. Yet, in the past decades, scientists have continuously challenged this mainstream theory by artificially lowering the water potential of the root bathing solution below that of the xylem vessels. And some of them were stunned by their observations because water was still being absorbed by roots uphill of both total and pressure potential gradients. And the cherry on top is that their observations also seemingly go against the second law of thermodynamics, which take this enigma to another level. How could we reconcile those observations with the laws of physics? Well, clearly we would need an active process that explains the water flow uphill of water potential gradients. However, we have two problems here. First, the process is not active solute loading in xylem vessels, as confirmed experimentally. And second, there is notoriously no active water transporter in plants. So we are left with a mind twister. My colleagues and I then decided to investigate those exciting enigmas with a multiscale model of the root hydraulics. This is a scheme of a single cell unit of the model where you can see cell walls in brown that are separated from the symplasts by cell membranes. Protoplasts are also connected by plasmodesmata. Water flow is pressure-driven in cell walls and plasmodesmata, while the osmotic potential comes into play across cell membranes. The next step was to generate the anatomical layout for the model. Digitizing root cross-section anatomies was really time-consuming, so then my colleagues Adrien Heymans and Guillaume Lobet had the idea to create the simulator Granar, which is a generator of any type of root anatomy. Granar is now online and open source, so feel free to try it. By combining the cell-scale hydraulic model to the root anatomical layout, we obtained MECHA. Here you can see simulated results of transmembrane water flow in blue from the cell walls into the protoplast, and then in red back from the protoplast into the cell walls. It happens at both apoplastic barrier location, here in the exodermis as well as in the endodermis. On the left, you can also see high water velocity in radial cell walls in the cortex, while here at the center of the stele, there's almost no water flux in cell walls, simply because all the water is already gone in the xylem vessels. Back to the simulation of our enigmas, we needed one last type of input data, the osmotic potential of each cell type. We found it in a treasure of experiments conducted by Enset al. They snap-froze roots during guttation and measured by electron microscopy the concentration in major solutes in xylem vessels as well as other cell types. Their findings were that the osmotic potential in xylem vessels was not enough to explain the intake of water, and that the osmotic potential in the cortex tended to be more negative than in the pericycle. That is all the information that we had when leaving on our quest for a new pumping mechanism for water. We decided to mimic the guttation experiment of Hens et al. By setting a higher xylem pressure than at the root surface and an osmotic potential that was more negative at the root surface than in xylem vessels. By doing so, we set both driving forces of water flow across root to push water from the xylem to the root surface. This condensed radial view tells us more about water pathways. The vertical lines show the limits between the different cell layers here in the cortex, and the gray bars show the locations of the apoplastic barriers, for instance here at the exodermis. The brown areas show the intensity of water flow in primary cell walls, and the blue areas show water flow in the simplest. Any transition between the brown area and the blue area relies on transmembrane water flow through aquaporins. Finally, areas above zero represent water flow that is inward, and below zero it is outward. Here we see some circumvention of water between the pericycle and the phloem sieve tubes, and some water leaving the cross-section through xylem vessels. The striking result here is that water flows from the root surface to xylem vessels, despite both driving forces pushing water out of the root. So the good news is that water flow uphill of water potential gradients is happening in both the model and the experiments. So we captured the water pump in the model, but the bad news is that we have no idea why. Now let us try to understand this water pumping mechanism by dismantling the system and building it back better. Here we have a system of 3 cells with both pressure and total water potential that are higher in the xylem than in the soil. Naturally, the total water potentials in the simplest are going to equilibrate to values in between those at the boundaries. Here on the transmembrane pathway, water naturally flows towards the soil due to the gradient of total water potential, while in the cell walls, water passively flows downhill of the pressure gradient, bypassing the different cell membranes due to the absence of apoplastic barrier. An important point is that given the osmotic potentials observed experimentally, the cortex is under higher pressure than the pericycle. By adding a lignified Casparian strip, we disrupt the pathway of water through the cell walls, but not the one through the membranes. Hence, water keeps flowing from xylem to soil at a lower rate. The next piece of the puzzle is the plasmodesmata. As you remember, water flow is pressure-driven across plasmodesmata, so that it will passively flow from the cortex that used to be under high pressure due to its high solute concentration to the endodermis and the pericycle. Because those 3 cells are now directly connected, part of the pressure of the cortex is lost to the pericycle, whose pressure and total water potential increase, while those of the cortex now decrease. This creates conditions that are favorable to the entry of water from the soil to the cortex across membranes and from the stele to the xylem, while across the endodermal membranes there's still an outflow of water. Depending on its boundary condition and hydraulic parameters, this system could either end up with inflow, outflow, or simple circumvention of water, just like in the MECHA drawing. From this point, how can we do better? Well, for instance, by adding a suberin layer on top of the endodermal membranes, because then that would block the backflow of water across those membranes. By analogy with the MECHA drawing, at small scale, water always flowed downhill of the relevant water potential gradients, but surprisingly, at the larger scale, when we take a step back, we realize that water overall flowed uphill of all components of water potentials from soil to xylem. So this solves the enigma of the uphill water flow, which comes with a cost because the root needs to maintain actively this gradient of osmotic potential across cells. We can also conclude that the regions of the root that are more mature and have suberin around their endodermis are able to pump more water. Now one last challenge. Find out if the impact of osmotic gradients in living cells is meant to be calculated by microscale models such as MECHA, or if simpler principles arise at a larger scale. When comparing root water uptake rates simulated by the microscale model, shown as symbols over here, Two predictions of the current paradigm of root radial water flow, shown as a gray line over there, we see that they only match when osmotic potentials are uniform within the root. Then, in order to find out what could be missing in the mainstream model, we derived analytical solutions of water flow equations in a simple triple cell system. It turned out that the mathematical solution took a simple upscaled form, with the driving force only slightly different from the one of the mainstream model, which only includes osmotic potentials at the root surface and in xylem vessels, while the upscaled form includes a totally new term, which accounts for the difference between osmotic potentials inside living cells on opposite sides of the endodermis. Luckily, we also found out that this simple upscaled model fully captures the impact of osmotic potential gradients simulated from the microscale in 2 or 3D roots under changing boundary conditions with a single set of parameters. Now for the conclusions, we have a new theory based on osmotic gradients within living cells, which may explain the long-lasting enigma of the uphill root water flow. We also found out that a simple solution of water flow equations across complex root hydraulic anatomies exists, and that it seemingly generalizes the current paradigm of root radial water flow, with the addition of this simple term here in yellow, which stands for the difference between average osmotic potentials in living cells on opposite sides of the endodermis. Among the next important steps come the quantitative validation of the impact of the water pump on stem water potential. Our first estimations suggest 0.2 to 0.4 megapascals difference in maize, yet further experimental verification will be required, for instance by monitoring soil and plant water status with in situ psychrometers, as well as cell-scale osmotic mapping with cutting-edge microspectroscopy techniques. Investigating if the water pumping mechanism plays a role in the avoidance of cavitation is another potentially important perspective. It will require the monitoring of root osmotic gradients' response to water deficit. And finally, it would be fascinating to study how roots maintain the osmotic potential gradients observed experimentally to keep the water pump working. Thank you very much for your attention. This work would not have been possible without the precious inputs from my co-authors, Adrien Heymans, Guillaume Lobet, Malcolm Bennett, and Xavier Drey. I would like to thank them, as well as the Belgian FNRS and the organizers of the SEB 2021 conference. Now I will be very happy to take your questions live or by email.